Thank you everybody for attending another Mission Bio office hour. Many of you might have already listened into the first session for primary data analysis using Tapestri Pipeline given by my coworker, Jose Jacob. And today I'm going to talk about, after you get the loom files, how you would like to perform the secondary data analysis using Tapestri Insights. It is part one of a three-part series. So today I'm just gonna give you an overview how to use Tapestri Insights software. The agenda for today composed of two parts. At first, I'm going to make a brief presentation on the benefit of using Tapestri Insights, what kind of input file you would need, and what kind of output files you can expect after using Tapestri Insights to perform the analysis. And second, and most importantly, I'm going to do a live demo on how to use Tapestri Insights to analyze real patient data. As you know, Mission Bio provides the complete bioinformatics solution from designing the panel all the way to secondary analysis. So Tapestri Insights serves as the last stop for you to perform the data analysis for data interpretation and visualization. So at the tool for visualization and interpretation, that Tapestri Insights is known to be very user-friendly. You can just download it for free from your Tapestri portal website. And it has this intuitive user interface, which allows you to analyze single sample as well as multiple sample at the same time. It also has the stringent QC built inside to allow you to remove the low frequency and low quality variants and cells. Plus, you could also import your own target of interest to perform the target analysis. In order to understand the variant and subclone to perform better selection and visualization, we also have tools inside for you to carefully review. We have the API-driven database engine, for example, ClinVar and Cosmic, that help you to interpret the data through annotation. You can also visualize your clone, your variant, in order to carefully select them. Lastly, we have a visualization tool for you to decipher the subclone architecture based on co-occurrence and the zygosity of the variants. So the input file for Tapestri Insights is cells.loom file. As you know that from last week's talk, it is the output file from Tapestri Pipeline. So it is essentially a customized annotated VCF file in .loom file format. It is compatible with Tapestri Insights. This is a sneak peek of the advanced filter that I just mentioned. It allows you to filter out cells and variants based on different quality metrics. I wouldn't spend too much time today on it since we will have a dedicated office hour on May 28th to talk about it. And this is an example output of Tapestri Insight you would expect. So on the left-hand side is the fish plots that allows you to observe the clonal size change over time. And on the right-hand side is the clonal phylogeny that you could construct if you have the inferred phylogeny. All right, without any further ado, let me perform the live demo here.
Yue, we had a quick question. Can Tapestri Insights be run on Mac, Windows, and Linux OS? And how much memory is recommended?
That's a great question. Yes, so we have a Tapestri Insight that can be run on both Windows and Mac. And I believe we require minimum 8 GB RAM. Can you see my screen?
Yes, it looks great.
Great. So this is, next I'm going to perform a live demo on how to use Tapestri Insights to perform the secondary analysis. As you can see here, I've already imported 3 files from, those are actually the files we collected from a real patient at different time points. You can easily add your files through click on the add here and import your loom files. So as you can see here, after you import your loom files, you could rename each of the file name to make them more biologically meaningful. You could also change the order for each of the sample in so that they can represent the true time point at which they were being collected. On the right-hand side, it is a performance matrix showing you the cell output, the total reads, and reads per cell. Those are no different from the output that you can observe from Tapestri Pipeline. But plus, Tapestri Insights also calculates reads per amplicon per cell, which is your true coverage. We recommend at least 30x, maximum 100x. If you over-sequence or under-sequence your sample, that may have an effect on your variant review process. Just keep that in mind. So after you import your files, they will automatically go through the advanced filter. As you can see here, we have 6 filters pre-built up to remove low quality genotypes, low frequency genotypes, as well as low quality frequency variants and the low quality cells. I won't spend too much time on explaining. Like I mentioned, we will have a dedicated session on it. Most of the time, you wouldn't need to change the advanced filters. Only occasionally, like if you have missing variants, you might need to change them carefully, and we would recommend that you work with a field application scientist or our support team member to perform the advanced filter adjustment. After they go through the advanced filtering, as you can see on the bottom, you have the low quality variants, low quality cells, and low frequency variants being removed. Ideally, we would like to see less than 10% low quality cells being removed. But for the other two, they do vary from time to time. And on the left-hand side, I just want to mention that you have the option of import your own target interest, what we call whitelist, in a BAT or CSV format. This allows you to include your variants for downstream analysis, regardless of the advanced filtering. After you remove those cells and variants, on the right top corner, as you can see, we have 29,399 cells left, which is the cells that sum of a total cell from the 3 samples after filtering, along with 24 variants. Okay, let's move on to the review variants tab and review the variants that passing the threshold. As you can see on this tab, you have 2 different sections. On the first section, you have your variant list. Those are the variants that pass the threshold of the advanced filter setting. And you have your subclone table that will be generated as you select the different variant. And let's first take a look at the variant table. So on your variant table, you have your variant name, and on the left side, you have this qualitative annotation for your variant. For example, how it affects the function, how this variant affected the protein change, and what kind of coding impact it has, along with what's very important, ClinVar annotation and Dan score. On the right-hand side, you have your quantitative data showing up for different variants. At first, you have the genotyped cells, which means how many percentage of the cells have been successfully genotyped among all the cells for this variant, specific variant. And the mutated cells, this is the percentage of the mutated cells in those genotyped cells. And on the last 3 columns, you can observe those quantitative data in different sample that allows you to make a judgment whether this variant changed dynamically in different sample at different time point, or they are just the same. So notably, by clicking on this wheel here, you can select to show or hide columns that you think most relevant to your variant review process. And you also have the opportunity to sort your variants by clicking on the inverted triangle on different columns. So in order to rank my variants for most important to less important, we usually recommend to rank them by clean-var and then. I won't spend too much time today on how we select variant, as we are going to have a more dedicated session next week on how to perform selection on variant and subclones.
Wow, Yue, this is really powerful. We have a question from an attendee. Is there a way to hide non-coding variants so you don't have to see a huge long list?
Great question. So like I mentioned that you can definitely trying to filter out your variants by clicking here. So non-coding. So I think for non-coding, we can actually we can actually trying to select here on the second column at the function. Like you can see that you can just select the coding variant here by unclick on other irrelevant variant that doesn't affect any coding regions. Okay, I will continue if there's no other questions. And like I mentioned, I have already sorted my variants by clicking on the clean-var and then next step, I would like to select my variants. So I select I have selected these 3 different variants. As you can see that after I select those variants, the subclone table is being populated automatically. And on your subclone table, let's take a close look at it. On each row, it shows the different variants that you have selected. And on the different column, it shows the subclone that has been constructed automatically based on the unique co-occurrence pattern of each variant. And those subclones ranked by their relative size. And on the right-hand side, as you can see here, that we have this subclone column. It's actually the clone size that's that make up less than 1% of the total cells. Actually, by clicking on the wheel on the right-hand side, you are able to choose whether to ignore or include the zygosity, and you can actually adjust the sensitivity in order to visualize the subclones passing certain threshold. By default, we set the threshold as 1%. And on the most right column, there is a column called missing genotype subclones. So those are subclones that lack such genotype on each row. And on the bottom, you also see that this dynamic change, or you see the different percentage of each subclone present in different samples, which allows you to compare. So this table, it includes both your true subclone as well as possible false positives. How do you distinguish that? It comes to the tool we call the variant viewer that allows you to do the selection here.
We just had a quick question about the subclone table. So someone noticed that wild type is the 4th column, but is it possible to put it in the 1st column position?
Great question. Yes, you can certainly do that. So those columns are also can be the position of the column can also be changeable. Like I mentioned, by default, they are ranked in a way that the size goes largest to smallest. Okay, let's go to the variant viewer tab. Like you can see here that the variant viewer allows you to visualize different variants in each sample, different subclones for their allele frequency, quality, and read depths in a violin plot format. So this allows you to visualize each single cells as well as how they present in the population wise in different samples and different subclones. So today I won't spend too much time on how to do the interpretation here. Again, this will be very important on how you can perform your subclone and variant selection in rationale. Lastly, let's come to this review subclone tab. This is the visualization tool that we have that available for you to visualize your subclone that you have selected. By clicking on show percentage, we normalize all of the samples total size into 100%. So as you can see, that bar plot allows you to both analyze single sample as well as multiple sample. And it shows the relative size of each subclone in different sample. So after you clicked on the show percentage, after the normalization, it also allows you to compare the clonal size across different samples. And the second tool we have available is fish plot. Fish plot allows you to observe the clonal dynamic change at different time. Like I mentioned, the reason we would like you to arrange your sample in a way that it represents the true longitudinal order is because this fish plot allows you to visualize them change at different time point. And one thing I would like to mention is that if you have your inferred phylogeny available, you could also use this tool to change the sample phylogeny. As you can see, as you change the different phylogeny order, it also automatically pop up the clonal phylogeny at the bottom. That's all for today. Hope you all learned something from how to use Tapestri Insights. I'm open to any questions.
Thank you so much, Yue. This was wonderful. So here's a question. You mentioned earlier on that you wanted to be between 30 and 100x sequencing, but what happens if you over-sequence? Do you have the potential to subsample, I don't know, that FASTQ or that loom file in order to get within the range? What would you recommend?
Yeah, great question. So for our platform, for data analysis, it's not necessary the more deep you sequence, the better. Actually, after you sequence, for example, over 100x coverage, you might be at risk of having a lot of false positives. So that will reflect in your review variance table. You might end up with a lot of false positives that you have to remove. And that could be mixed up with your true high quality somatic variants. And a way to trying to maybe to save this situation is that you could downsample you could downsize your sample so that you don't have that deep sequencing data there.
Great. Another question. Do you recommend analyzing samples from different patients together?
So I would recommend to analyze them separately. Like I mentioned, this allows you to analyze the same patient sample at different time points so that you can sort of observe the change. But if you have different patient sample, then I would recommend analyze one at a time.
Good to know. And then a couple questions here about exporting data. Is this data exportable?
Yeah, great question. Apologies, I forgot to show that. So on this page, as you can see, we have this export button. We could actually export the filtered data into a report that allows you to either for your publication purpose or your patient report.
Wow. And then is the exported data always the filtered data? What if I wanted all of the raw data before filtering? Is that possible?
Yes, great question. So you could also click on here. as you can see here, that on each tab we have all of the export data. So on the first tab, if you click on export filter data, this is kind of sort of your raw data that you have, but passing the advanced filtering. And on the second tab, you could export your data, also filter, including your select variant and the subclone that you constructed. So for the raw data, as I mentioned, you have already get it from Tapestri Pipeline, which includes your loom file and some other, for example, BAM and VCF. Those can all be used for you to perform any different analysis if you have, for example, some other R package available.
That's great. And then, so with this Tapestri Insights software, are the images good enough to go right into publications?
Yes. So actually, we have already collaborated with many researchers. They have been using this tool to publish data for using, either fish plot or our clonal phylogeny tree to be included in their publication. So what I can do is I can do a quick export for you, just to for you to get a general idea how it would look like. For example, this is the export of the for the analysis that I just performed. As you can see here, we have this high resolution fish plot and clonal phylogeny that you can include for your publication.
Wow, this is great. I mean, it also seems it'd be really nice for reporting purposes for clinical samples and for records.
Exactly, yes.
Great. I think you might have answered this, but we'll ask it again. Can I also export figures individually as opposed to having it on this report?
Great question. Yes. Here, as you can see, by clicking on the export, you can either maybe if you are just interested in the fish plot, you could just export the fish plot. Or if you're interested, just export the subclone table. Also, you are able to do that. And same here. If you're only interested in export your bar plot, there is a function that you're able to use at the export part button. Just select the bar plot. It will just export that for your publication purpose.
All right. And can you change the colors of these bars?
Nice question. I believe that you can. But this is a question maybe we can talk about further offline. Apologies that I'm not quite, I don't really know. But I do know that we noticed some other scientists that have already, that they used our figure that were able to change the color successfully.
No problem. I'm sure we can find that information. Okay, well, if there are no more questions, this was a wonderful introduction on Tapestri Insights software, and I'm really looking forward to your part 2 and 3 to go more in depth about the biology and how you select variants and filter them. So Yue Wang, thank you so much for giving us part 1, and we look forward to the next parts.
Thank you, Kelly. Thank you, everyone.